Good evening, everyone, and good morning to Australia and Canada. My name is Mr. Adnan. I'm a consultant colorectal surgeon in the UK, and I'll be your host today for this webinar. We have two world experts joining us today. Professor Georgina Hold, who is the gut health professor in Sydney, and she is a well-known figure for gut microbiology. I also have Dr. Josbert Keller, who is the consultant gastroenterologist in Netherlands. He's a co-founder for Stool Bank in Netherlands. Welcome to you both. Before we move on, I'd like to say thank you to all the participants who joined in. Without you, it would not be possible. Please stay online and you will learn from this webinar. I also like to introduce you about the format of this webinar. Our two world experts would be giving a presentation first, which is about 5 to 10 minutes presentation, and followed up will we have questions from you, and we will try to create a discussion here. So you can type in, as you can see on your right-hand side screen, all your questions. If you would like to discuss amongst yourself or address to other participants, you can type in everybody. And if you want to send a specific question to moderator or to our experts, you can select by selecting moderator only. So I would like to start with Professor Hold, who will give us some idea about the gut microbiota and gut health. Over to Georgina.
Thank you so much, Imran, and good morning or good afternoon, everybody. Isn't this exciting? I am so excited to be here and to give you some information about the gut microbiota, and then Josbert will take over and talk more about FMT. So we know there's so much information out there about the gut microbiome or the gut microbiota and its role in health and disease. It's probably the most exciting paradigm shift in medicine in the last sort of 10, 15 years. Now, why do we get so excited? Why do we think that we can do so much with this? Well, there's so many microbes associated with us as humans. If you look on a cellular basis, the number of human cells is about 30 trillion, and there's around 39 trillion microbial cells. So just on a cell per cell basis, they equal the number of human cells. So we're carrying around as many microbial cells, if not more microbial cells, than human cells. If we go a level deeper than that and look at the genetic level, you add up all the numbers of genes that we have in our human genome, which is around 20,000, but then add up all the number of genes that we're carrying from our microbial genome, it's around 2 to 12 million. So if you look on that basis, actually we're carrying around only 1% of human genetic information compared to the rest being microbial. So it has to be able to do so much to influence our health and disease. So if we look at the microbes that associate with us, we find some very interesting facts, which makes sense, is that every part of our body has a different kind of physiological environment. And so it attracts different types of microbes. And so we have body site specific composition. And wherever you are in the world, in general, the body site composition remains relatively similar if you talk about the types of microbes that like to associate with that type of environment. But the microbes are hugely influenced by what we eat, what our lifestyle is. So we do see changes, and geography is a very important driver of microbial composition. One of the things which has become clear to us is that having lots of different microbes in a particular environment, having a rich diversity, rather than looking at the total numbers, it's about variety, variety being the spice of life. Diversity is generally a marker of health. So if you have lots of different types of bacteria, in general, that's a healthier place or a healthier ecosystem to have than something that is driven by just a single or a few microbial species that are there in a high abundance. One of these words that we always talk about in relation to the microbiota is dysbiosis. Now, what does that mean? So dysbiosis essentially means a change from what is normal. So if you look on the slide at the side over here, what we have is a seesaw, a balance. And what we have is a balance of different microbes. Over here, we have symbionts. They're the microbes that live associated with us, that do some health benefits for us, maybe don't do on their own, they maybe don't do too much, but between themselves, they do very good things for us. We have commensals. Commensals are also good guys. Commensals are part of the healthy microbiota as well. But we also do have this balance of pathobians, the not so good guys. And so that balance between good guys and bad guys puts us in a normal, healthy state. What happens in relation to diseases or to infections or health conditions is this balance goes out of kilter a bit. So we have maybe an excess of pathobians, and so the balance tips in the wrong way. Now, this would be defined as dysbiosis. Dysbiosis can mean a difference in numbers. It can mean a difference in the microbial functioning capacity. Now, what do we know about dysbiosis and health? So we know that changes in the microbiota, which is what I've told you dysbiosis is associated with a number of different conditions, whether it be atherosclerosis, obesity or metabolic disorders, very many different types of cancers, both hematological and solid cancers, solid tumours, neurological disorders, liver disease, diabetes, maternal health and pregnancy, and even something as obscure to microbial language as joint pain. And from my particular interest, and probably what we're going to talk about today, is about its role in inflammatory bowel disease and irritable bowel syndrome. All of these conditions have been shown to have a difference in changes in microbiota. However, the differences are disease-specific in some cases. So what does this dysbiosis mean in the context of disease and health? Well, it can mean reduced diversity, loss of richness. It can mean a difference in function. So if you change the numbers of the types of bacteria there, they're going to do different functions as a microbial community. So it could be a reduction in healthy metabolites such as short-chain fatty acids. It could also be not just bacteria, it's also the fungal and the viral component. And we do get very lazy about talking about those two parts of the microbiota. But the microbiota is a collection of not just bacteria, but also viruses and fungi. And when we do talk about the bacterial component, the alteration in the two major groups, the two major phyla, Firmicutes and Bacteroides, that's what we really look at again as a marker of changes in health. But that doesn't give you specific species changes. And so this slide, which is very similar to the one before, was just indicating that not only do microbes change in the context of disease, the disease itself also changes the microbes. So this is a two-way relationship, and we have to remember that as well. So in regards to fecal microbial transplantation, how does that fit with what I've just been talking about? Well, there are lots of ways that we can target dysbiosis, and it depends what the context is. Some of these approaches are quite imprecise. Some of them are incredibly precise, and depending on what the question is that you want to ask or the condition that you want to impact on, depends on the type of approach that you're going to use. So when we talk about fecal microbiota transplantation, we're really talking about replacing or restoring the whole microbial environment within a particular location. So this is relatively crude. Currently, with current approaches, it is a relatively crude, restoring everything approach. But as we move on and we talk more about FMT when Josbert takes over, then really we're going to talk to you as well about how FMT is moving into the next generation of treatments. And so I think I hand over to Josbert at that point.
Thank you, Georgina, for this excellent introduction about the challenge of the future years where we should target dysbiosis. And so far, dysbiosis can only truly be targeted in a way that it's clinical relevant with fecal microbiota transplantation. This was first described by Eisenman, who was a US surgeon who treated several patients with severe enterocolitis caused by antibiotics. We didn't know about Clostridium difficile at that time, successfully with enemas containing donor feces suspension. And interestingly, it lasted until 2013 before the first randomized trial showed that indeed infusion of donor feces, fecal microbiota transplantation, of healthy donor feces, I should say, is much more effective in curing patients with recurrent Clostridium difficile infection compared to the standard of care at that time, which was vancomycin. And after publication of this randomized controlled trial in 2013, FMT was also incorporated in the guidelines for treatment of recurrent Clostridium difficile infection. So how does fecal microbiota transplantation work? In fact, doing the transplantation itself, that is very easy, and every skilled gastroenterologist is able to perform this in a safe way. You can infuse the donor feces through a nasal-duodenal tube in the small bowel or even in the stomach if you add PPIs, or you can infuse it through a colonoscope in the colon, as you can see here, or, and that is also something which will come faster than we all believe, you can just give capsules to patients. And this also appears to work easily. So this is the easiest part. The more difficult part is the process of screening and selection of a donor, the process of making a suspension for FMT and storage, and of course the indication. So screening and selection of a donor, which appears quite easy, is even more difficult than finding a donor for example, a blood transfusion, because besides that we screen very carefully for the presence of potentially transmittable pathogens, we also select donors that have a very low risk for disorders that are associated with a disturbed microbiota. And therefore, only about 5% of potential donors are suitable to become a feces donor in practice. Initially, people used fresh donor stool for FMT, and that was truly a logistic nightmare, as is illustrated by this cartoon that was published together with our randomized controlled trial that was published in 2013. And I think that all physicians who worked with fresh donor stool will recognize the problems that were encountered at that time. And in this regard, it was very important that Dr. Lee and colleagues from Canada were the first to publish the finding or the results of a study that compared the use of fresh and frozen donor stool. And indeed, both were equally effective in curing CDI, I should say, so in curing Clostridium difficile. Whether it is equally effective for other disorders, and we will talk about that later, we don't know yet. But this observation really made it possible to start storing feces suspensions for FMT and sending them out or using them if a patient needed it. So that enables us to initiate stool banks. And for example, in the Netherlands, we have the Netherlands Donor Feces Bank, which is a national entity and sends out suspensions to all hospitals where patients with recurrent Clostridium difficile are treated. A stool bank could be national level, could be even work at an international level, or could only work as an institution-based stool bank for an expert center. And it is important to realize that all those stool banks should fulfill the same quality standards. In the beginning of FMT, everybody had his own protocol, but now we are trying to standardize the process of donor screening, of processing of donor stool, and stool banking. And there are several attempts to standardize this, and one was by Camerota, who chaired a huge international group of experts, and recently we published this report in Gut, and that was really an important landmark again. And there's also an UEG supported project to develop a kind of model for a European stool bank, and we hope that we can publish that soon as well. So if people are willing to establish their own stool banks, they can just go through those manuscripts and try to figure out how to do it. It will be very easy, but it's again very important that everybody fulfills the quality standards that are required for this treatment. Which patients do benefit from FMT? Well, currently only for recurrent Clostridium difficile, FMT is mentioned in the guidelines, and there it is very successful. It's easy to perform. With one single infusion, you can cure more than 80% of patients. However, it is a very promising treatment approach it might be a very promising treatment approach, I should say, for many other disorders, in particular for ulcerative colitis, irritable bowel syndrome, and hepatic encephalopathy. There are already randomized controlled trials that indeed show that there is some effectivity, although, for example, in irritable bowel syndrome, results are not consistent yet. And there are many questions to be addressed before we can really incorporate FMT in daily clinical practice for those patients. This picture shows that for ulcerative colitis, we are almost there. I think that there are 4 randomized controlled trials so far published that all show an effect, although it was not significant in the study from Amsterdam. But if you pull those data, there is indeed a clinical efficacy, about 15 to 20% additional responders compared to the placebo group. However, there are still many questions to be addressed before we can really consider this as a kind of standard treatment approach. We just don't know which patients we should select for FMT, which ulcerative colitis patients. We don't know what is the best timing for FMT in those patients. We don't know what is the treatment regimen, how should they be pre-treated. So many questions to be answered before we can really offer this in daily practice to our patients. But as you can see, it is very promising. So is it safe, FMT? I think this picture shows that it is safe. This is adapted from a large meta-analysis incorporating 135 studies where side effects were reported. And as you can see, serious side effects are very rare and even mainly related to the procedure, like aspiration for upper GI, perforation for lower GI, delivery by colonoscopy. And I don't think that there are true serious side effects except for sepsis reported caused by the donor feces suspension itself. So what do we see as side effects? They are mainly mild and transient. The GI side effects like diarrhea, which is normal after pre-treatment with bowel lavage, of course, but there's also abdominal bloating. Some patients report constipation, and some patients report fever, which is 2% in patients in general. And it's interesting that it's about 8%, fever is encountered in about 8% of patients with active IBD who are treated with FMT. So the true question is, are there also other diseases that can be caused by FMT? So some reports suggest that there might be a flare of IBD after FMT, which is very low. The likelihood is very low, but we don't know if you can also induce other disorders like, for example, multiple sclerosis or autoimmune disorders, and we really need long-term follow-up in a structured way to answer all those questions. That brings me to the next slide.
So what I hope we've managed to provide you some information on here is that FMT is an established treatment for recurrent Clostridioides difficile infection, that the safety and efficacy is it's an easy approach to use, but that donor screening and selection of donors and preparation of FMT and the use of stool banks is a critical step to allow us to make this a more standardized approach. And where we are in terms of emerging treatments, we still have a lot to learn, but we have a good science basis from the recurrent C. diff literature to allow us to move forward. I'll hand back to Imran, the moderator, now.
Mm-hmm. I can see there's questions coming in, and I would like to continue with Professor Hold. The question is, can we all put together one consensus? Is FMT a tool? Is it a drug? Is it an organ, or is it a cell transplant?
Well, for a starting question, that's kind of a big one, isn't it, really? So it really, I think actually, in the interest of knowledge, I'm really going to hand this one over to Josbert. I hope you don't mind, but I think Josbert is much better placed to answer that than me.
Well, Imran, I think there is debate about this because regulatory authorities just don't know how to regulate this. And in several countries, and it's difficult to imagine, they have chosen to make it a drug. However, I think that all experts in Europe agree, and we have recently published a letter about this, that as long as it is just feces and it is voluntary donated feces, we should consider it as a transplant product. There is no true difference between, for example, the whole process of blood donation and treating a patient and feces donation and treating a patient. As soon as you are going to enrich the feces, not process it in a way to contain the community or the bacterial community as a whole, but as soon as you're going to enrich it and choose several pathogens or then I think it is a drug and it should be regulated as a drug. So a lot has to be done to convince the authorities about this, but I think that in the FMT experts all agree about this. So it should be a transplantation product.
Okay, thank you, Josbert. That's a very clear answer. Georgina, the questions coming in. So I can see dysbiosis causing a lot of diseases, the cancers, metabolic disorders, IBS, functional disorders. Where do you see the future of FMT here? You also mentioned it's not a very precise treatment. Can we not just select some of the bacteria or bugs or viruses and do an injection or treatment, so a more specific treatment for these conditions?
So again, a very, very important question, one which as a research community, we kind of scratch our heads and think about a lot. So the point in terms of, can we not just take the good bugs and put them in and that will solve everything? The issue is that the good bugs are different between individuals. We each have a different consortia of microbes that lives in our gut. So one approach for somebody may not be right for somebody else. So that's the first thing to say. And also, because the microbes that live in your gut or that live in any community are a community in their own right. And so they take from each other, they give to each other. So it's about the whole community of microbes and what that is imparting to us as a host in terms of metabolite production. And so it's really not quite as simple as fixing one bit. Because all you'll do is move, you'll just alter the dynamics of the community. Even if you think you're putting loads of good bugs in, you'll alter the dynamics of that community to such that it's not the community that you're even trying to be anymore. So we do need to get more precise FMT approaches, and that certainly is something that is being done. Whether it will still be a fecal microbial transplant and whether it will just be a microbial transplant at that point is where we are at the moment. And as Josbert says, that then takes FMT out of this transplant into a more precision treatment. And so we're then entering a very different sphere, and particularly in terms of legislation.
Extending on to your question, Professor Hold, I just have another question. If I have a fecal transplant, can you treat my obesity?
Can I treat what, sorry?
Obesity.
Obesity, sorry, I heard wisdom teeth. I thought, gosh, that's a new one. Can we, well, so there's some very, very interesting data that's come out. There's a lot of animal data to show that you can induce obesity by altering microbes, and then that you can actually get some benefit for treating obesity-associated disorders. But again, some of the most promising work in relation to that is to do with using directed specific strains. So, and that's because the FMT approach is giving us good information, but then we need, as diseases such as obesity-associated disorders and inflammatory bowel disease, they're very complex diseases. We have to learn as we're going along. So there is work being done in that sphere in terms of trying to microbially manipulate to improve obesity-associated disorders, yes.
Okay, thank you, Professor Hall. This very clear answer. I have a question to Josbert now. This question been asked about use of colonoscopy in terms of infusion of FMT. Can you enlighten us in terms of different?
Sorry, Imran. Imran, could you repeat the question for me?
Can you enlighten us on different routes of FMT infusion, especially where it should be infused?
Yes, I saw this question also be asked via the chat box, but I think that at this moment we don't know whether colonoscopy or upper GI delivery through a nasal tube is more effective or more dangerous exactly. There's no head-to-head comparison. We know that both are effective for treating recurrent Clostridium difficile infection, and that the suggestion that colonoscopy is more effective is not based on large numbers. So I think that you can just do what you are used to do. And for other disorders, it may be different, and we just don't know yet. We hope that capsules will become available in the near future. And if it is indeed shown that you can treat C. diff, recurrent C. diff, with, for example, only 20 grams of feces, then it will be possible to encapsulate this in a reasonable number of capsules that can be swallowed by all patients. And that would be the best for patients because it's just less demanding. So let's hope that that's coming soon.
So as the questions are coming, I think what are the FMT? What are the borderline indications in research where we can actually go into with FMT?
Imran, I missed you for a
Sorry,
second
Josbert.
again.
So as the questions coming, people like to know what are the clear indications for FMT? Where else we can use FMT for treating other conditions? And in future, what will be the prospect like? Can be treated for alcoholic hepatitis or another metabolic conditions?
Okay, so I'm going back to the slide about FMT and other disorders. I think that the only clear indication at this moment is recurrent Clostridium difficile infection. And it's also important to mention this because if you offer it to other patients, it's more or less experimental. So for ulcerative colitis, I believe that we are almost ready to offer this to certain patients. And there's also the question of the president of the UEG about Crohn's disease. Well, there is a lot less data in Crohn's disease compared to ulcerative colitis, but I think that there's also some indication that it might work. And in particular, patients with Crohn's disease are desperately waiting for more friendly, less toxic, and more effective treatment approach. And so I think really that it should be considered also in studies in the near future. And maybe we need to pre-treat patients with, for example, prednisolone before we offer FMT in those patients, but we just don't know, and a lot of work has to be done. So in irritable bowel syndrome, it's also a very interesting question because some studies don't show any effect. some do. And recently it was shown in a huge study from Scandinavia that if you use one that should have been a super donor, you could really cure many of the patients with FMT. So a lot of work also has to be done, but these are all indications where I believe that we will be in a certain way successful in the future in offering it to a subset of patients. Does that answer your question? Or would you like me to talk also about microscopic colitis, metabolic syndrome, graft versus host disease, because it is now offered to many, many patients?
Okay. Now, coming to Professor Hold, obviously we are reading a lot this time about coronavirus. Can this be FMT be a way forward treating coronavirus?
So I think there's two things that I would there's another point that I would like to raise within the discussion in relation to coronavirus, and that's the fact that we need to think about the implications of coronavirus infection in the science of FMT, and that we need to make sure, because of the paper that was published last week showing that that coronavirus can be detected in stool, then there's also obviously the implications of making sure that FMT products are not contaminated with coronavirus as well. So I think that's something really important that we, as the science field, needs to think about and particularly consider in relation to preparation of FMT. In terms of can we cure coronavirus with FMT, it's not a disease which is impacting particularly on the gastrointestinal tract, so it's not really going to be a useful way, or it's not a useful way forward for that. I don't have, Josbert, would you add anything to that?
Well, I read a report of a Chinese physician, of a group of Chinese physicians treating patients with corona disease, and they mentioned that they offered also prebiotics and probiotics as a kind of supportive care, but there's indeed no evidence at all, and I don't think that there's any evidence that you can treat a virus in the bowel or in the intestines with FMT. But it is also, in fact, asking the question whether you could treat coronavirus with FMT. That's also the question: shouldn't we offer FMT to all patients if there is no other treatment available? And the answer is definitely no, I think. And in particular, also in severely ill patients, I think we should be very careful with FMT. We've also seen one probiotic study in pancreatitis patients showing more mortality in those treated with probiotics compared to placebo. So it is not certainly always safe. So we shouldn't offer it if there's no evidence at all. That brings us also to the question, who could benefit from it? And I think we need criteria for kind of compassionate use of FMT. And at least one of the criteria should be, is there an indication that dysbiosis of the microbiota is involved in the pathogenesis of the disorder? And another criterion should be, is there another treatment approach that is still available, because FMT is experimental in those patients, certainly. And finally, you should always consider, is it safe in those particular patients? So there are a lot of things to consider, and I think we should also at a certain moment come up as experts with a kind of compassionate use criteria, but that's just not available yet.
Can I pick up on what are the criteria to use for treating FMT? Again, I go back to the point that the majority of the data that we have in relation to FMT and how well donor feces is transplanted into a recipient, we really only have most information on what's going on the bacterial side of things. There is some information coming around what changes are happening in the viral component, because obviously you're transferring viruses as well as bacteria. And so we need to also understand that more before we can understand how best to, what other indications we can use FMT for. And I see a question coming in about the use of FMT in the context of cancer and immunotherapy, and should FMT be used as an adjunct? And so I mean, there's some really interesting information out there showing that microbial communities in a patient have a huge influence on immunotherapy outcomes and the efficacy of the treatment. And so there are some studies out there looking to modulate the microbiota of an individual prior to immunotherapy. So I think, you know, there is space within that area to certainly improve immunotherapy and potentially other therapeutic approaches to and maybe FMT can fill that space. So I think that there's lots of considerations.
Yes, I completely agree with you.
Josbert, I have a question for you. How we can set up FMT services or FMT related stool banks in Eastern European countries or the countries that do not have these facilities?
Well, I think to facilitate the foundation or the initiation of new stool banks over in all countries in Europe, we are working now on a kind of model, and that is really how to set up a stool bank, and it will even provide a kind of working plan. It will also contain standard operational procedures for the main steps in the process of donor screening, processing, and storage of feces. And people who are willing to do so are also welcome to join the UEG working group on FMT, on fecal microbiota transplantation, which is hosted by the European Helicobacter Microbiota Study Group. So if people are willing to join this because they want to set up a stool bank, they are welcome in our working group, and everybody is willing to share his their experiences with them.
Extending on this one, Josbert, do you have any consensus guidelines or recommendations which we should follow in Europe or in Australia or in France?
Yes, as I mentioned, there is a consensus report published. It was chaired by Camarota from Rome, who did a really good job, and it is published in Gut last year. And there will be a new kind of manuscript about this stool bank model, and we hope that it will be published forthcoming, let's say, months somewhere. And we are working on that. So but there is already one consensus report, and if someone needs it now, he can also contact me, and I can share many of the information already. Excellent,
thank you, Professor Hold. We're coming back to dysbiosis here. There's quite a few questions. Are we really handling a dysbiosis by FMT? Is that the change of cells, bacteria, which we're introducing by FMT, or is it just a self-dysbiosis to correct by?
So it's a really interesting question, and there's not one shoe that fits all. So if you look at, in relation to FMT, if you look at recipients who get the most benefit from FMT, then in general, they show a much stronger engraftment of the donor's microbiota. But again, it comes back to the question that you're trying to answer with FMT. So when we have Clostridium difficile infection or Clostridioides difficile infection, what we've essentially got is an overgrowth of one species, Clostridioides difficile. So anything almost that you can boot that infection out with will be an improvement. When you then come to the more sort of complex diseases such as IBD, we're not looking to boot one single sort of pathogen out. It's a field defect. And so again, it's about working out what the field defect is and being able to restore that. And the two difficulties with that are, one, in general, we didn't know what the patient's microbiota consortia was before they got ill, because we don't routinely monitor an individual's microbial consortia. And two, we don't understand, it's different for different individuals. We haven't got a specific signature that says this organism is the problem in IBD. if we did have that, it would be we would know how to fix things, but we don't. So we're working with not a complete knowledge base at the moment.
Thank you. Coming back to Josbert, I need a little bit more clarity in terms of FMT procedure protocol, and some of these questions will be answered on those. How do you select your donors? How you address a concept of super donor, and how many infusions you plan to give to a patient?
Well, we select our, well, we have a lot of potential donors, and we select our donors with a very long questionnaire. As I told before, it is more extensive than the blood bank does. And it also looks at weight. For example, your BMI should not be above 25. You shouldn't have first-degree relatives with ulcerative colitis. Well, there are many more questions than are required for blood donation. Then what is the other question is about the super donor. Well, yeah. So we for a Clostridium difficile, there's no super donor needed at all, we believe. Well, there's probably a difference, but the efficacy is that high that it's very difficult to show any difference between donors. But for other disorders like IBD, there is certainly a need for super donors, and probably also for IBS, and maybe for hepatic encephalopathy. And we don't, just to be honest, we don't really know at this moment how we can select super donors. So we use a test to see whether there's Treg induction by microbiota of certain donors for a study that where we apply FMT for ulcerative colitis patients. But that is very difficult, very experimental, and it's just not ready for clinical use yet. But I think that is really the challenge for the forthcoming year, finding the super donors and then making from these super donors, let's say, targeted or specific microbiota therapeutics that are not anymore dependent on the donor itself. And then the question is how often should we? Well, this is also a question that I saw asked by one of the people in the audience. How often should you repeat FMT? And that's only once for C. diff. Sometimes you have to repeat it, but a post-FMT relapse can easily be treated with, for example, fidaxomicin in many patients. So for C. diff once, for ulcerative colitis, for example, I think that 2 FMTs is a little too few. But we don't know how many are needed exactly. There was also an Australian study in which 40 enemas were given in a row, and I think that might be too much. But somewhere, let's say 4 or 5, and maybe we have to repeat it at a certain moment. We have to study this still. There was also one question, Imran, that I would like to address, whether you need antibiotics as pre-treatment if you treat patients without Clostridium difficile with FMT. And I think that the answer is yes, because we know, well, it's not published, but in healthy volunteers, if you do microbiota therapy, you'll have more engraftment after vancomycin pre-treatment for several days than if you don't do pre-treatment with vancomycin. So you need to create a kind of niche where the healthy microbiota can engraft. We don't know what is the best way to do this, but there's certainly something needed as pre-treatment in patients without C. diff as well. Do you agree, Georgina?
I do. I absolutely do agree. Yes, I think that, you know, we don't know what it is about the vancomycin treatment that is making the engraftment better. Is it the reduction in the bacteria? Is it the altering of the viral component? Because once you alter the bacteria, because a lot of viruses host is actually bacteria, it could be a modulation of the viral component as well. We just don't know at the moment.
Okay. Georgina, if you can extend on, obviously antibiotics making us feeling sick now, and we need to take our probiotics on a regular basis. If you are prescribing antibiotics to your patient, do you normally prescribe probiotics with them?
Well, I don't prescribe anything, so because I'm not a clinician. But I think your question is, should we be using alternatives to antibiotics? What do people do in terms of probiotics? I think that there's a lot of. I think we're kind of getting to an age where the new age probiotics are going to start appearing soon. I think probiotics is going through a sort of a rebranding at the moment, where we're starting to look for potentially more functionally useful microbes than based on the science that's coming out. We're able to be working with the more tricky organisms that before we couldn't keep alive, and we can actually maintain their viability in probiotic cultures now. So I think probiotics is really going through this kind of change, and I think that there's a lot of exciting research to be done. Josbert, would you agree?
Yes, I completely agree. But we don't prescribe probiotics in the Netherlands, almost never. But it's really something to work on.
Okay. Obviously, I'm extending on to your procedural protocol here. Do we have any long-term data on safety and efficacy of FMT?
Yes. Well, we have limited long-term data. We recently looked, for example, but there's no clue or no indication that it is really dangerous on the long term, also not in with regard to, for example, autoimmune disorders or obesity. There's one case report suggesting that obesity is induced by FMT, but I don't think that was very convincing. I do agree that we should be careful, but I don't think that there's any data that it is dangerous. Well, we need long-term data. For example, but the problem, for example, for Clostridium difficile, you don't have to worry too much, because this mainly affects older people well, many of the patients that we treated in our initial trial have died due to other disorders in the past 10 years or 8 till 10 years. So for those patients, I don't really worry about long-term consequences. But for pediatric indications, for IBD, and those younger people, we really need long-term data. And we are working now on a European kind of registry where it will also be a model, because I think every country has to do it by himself. But we will provide the European registry where we can just store all those data, and we can follow then really the long-term side effects of what we are doing.
Thank you, Dr. Keller. At this stage, I'd like to ask Professor Hold to give her concluding remarks about FMT and fecal microbiota, and then we will conclude this session.
I think that it's been absolutely amazing, the opportunity to have this webinar, and thanks so much to UEG for having the insight to do that. I hope all of you that are listening have enjoyed and learned something. So we've talked a lot about what we do and don't know in relation to FMT. There definitely are unresolved questions. There are ways in which things are going to change in the future. So we've touched a little bit on about how much FMT is needed, how often it's needed, how much of starting material is needed, and so whether encapsulation and how the preparation of FMT needs to move forward to become both sort of socially acceptable, but also a more cost-effective way of delivering FMT. So encapsulation and lyophilization of the encapsulated material is definitely something that is becoming more and more common. Josbert talked very much about the need to carefully select donors and the criteria needed to select donors. Questions around how often should you be screening the donor, because life factors are going to have a huge impact on microbial communities, and it does take a while to screen donors. but how often should that be done once they're into the program, once they're accepted? Should they be screened every 6 months? Should they be screened every time they provide a sample? This is all what needs to be regulated. When should we treat people with FMT? Should we be pre-treating them with antibiotics, or should we be pre-treating patients who are going on to immunotherapy with FMT? We're still learning what is the right and wrong ways to go about these things, but it's certainly a dialogue that is now open. How should we process the FMT? There's a lot of talk now about anaerobic processing. So most of the bugs in the gut actually are anaerobes. They don't like oxygen. The gut is a closed system, really. So they survive in the gut with no oxygen. So we should potentially be paying more attention to processing them in that way as well, because we know if you put anaerobic bacteria into an oxygen, then they change their metabolism. So there's a lot of thoughts about, is that why FMT is not so effective in, for example, IBD compared to Clostridium difficile? Is that because we're not, the microbes are not as effective for treating IBD because they're doing something different? And then we've also touched on the legislation. Is FMT a therapeutic? Is it a transplant material? Does it require additional legislation as a drug? These are all questions that we are grappling with, and we are, as a field, moving forward. And I think there's a lot of interesting work being done in Europe. I think we are definitely leading the way, and we should be very proud of that. And it's due to the efforts of people like Josbert who are helping us to stay there and be a field force to be reckoned with. And that's about all I have to say.
Professor Hold. Dr. Keller, if more want to get or the
sorry, Imran. Sorry, Imran. The connection failed again. Can you repeat the last?
Dr. Keller, if one of our participants want to contact you or seek more information or clarity, how they can contact you on this topic, and where they can find more information about this topic?
Well, please send me an email. And again, I would like to refer to the interesting report of Dr. Camarota and our forthcoming manuscript about stool banking. But you can send me an email if I can help you for setting up a stool bank or for any questions.
Okay. Thank you very much, participants. Thank you, experts. I'd like to conclude the session today. It has been extraordinary effort behind this webinar, went from UEG Education team. Thank you, UEG Education team and the Education Committee for this opportunity. And we are aiming to have further 2 webinars this year. And if you want to stay tuned, our next topic is artificial intelligence and its role in gut diseases. And hopefully you will all come back to us for the next time. Thank you, good evening and good night.
Thank you, Imran.
Thank you, bye-bye.
Thank you, bye.